This experiment tests the ability of Staphylococcus aureus to utilize human hemoglobin as a sole iron source. First, isolate red blood cells from fresh human blood. Purify the hemoglobin by high-performance liquid chromatography to ensure the integrity of hemoglobin. Now, add hemoglobin into iron-depleted medium to supply the iron in the form of hemoglobin. Next, culture S. aureus in the presence of hemoglobin and measure its ability to utilize hemoglobin as an iron source. The results show that S. aureus proliferates in the presence of hemoglobin as a sole iron source.
This method can help answer key questions in the field of microbiology, such as whether a pathogen can utilize host proteins as sources of iron and which mechanisms are required to utilize iron using this process.
Though this method can provide insight into iron acquisition of Staph aureus from hemoglobin, it can also be applied to the studies of other bacteria and how they use iron-containing proteins from the host.
Demonstrating this technique will be Gleb Pishchany and Kathryn Haley from my laboratory.
First, obtain freshly isolated human blood supplemented with an anticoagulant and maintain at 4 degrees Celsius throughout the purification. Pellet the red blood cells by centrifugation for 20 minutes at 15,000 Gs. Carefully aspirate the supernatant and gently resuspend the pellet in ice-cold 0.9% sodium chloride solution. After 3 washes, resuspend the pellet in 1 volume of ice-cold 10 mmol tris HCl to lyse the RBCs by osmotic pressure. Then add toluene to 20% final volume and incubate at 4 degrees Celsius on a rotator overnight. Next, centrifuge the lysate at 20,000 Gs for 1 hour. Collect the middle hemolysate fraction, leaving the upper toluene layer and the pellet undisturbed.